Good afternoon, everyone. We're Team ePatch, and we're excited to share a new technology in the space of mRNA and DNA vaccine delivery. To briefly introduce ourselves, my name is Garv. I'm a student at Georgia Tech with a background in vaccine development and delivery. Cindy Sindel is a principal at Georgia Tech Venture Lab. Todd Bhamla is an assistant professor in chemical engineering at Georgia Tech. And Mark Prausnitz is a Regents professor in chemical engineering at Georgia Tech. I'll begin by speaking a little bit of a few patient stories. Luke Hutchinson is a Moderna clinical trial participant, and after receiving his shots, he experienced bone and muscle aches with a high degree fever, and he had severe adverse effects. And his quote is, "Nobody prepared me for the severity of this." Dr. Hussein, after receiving his shots, went into anaphylactic shock and had to self-administer an EpiPen as a result. And these severe adverse effects have contributed to the high vaccine hesitancy rate, not just among the US population, but even amongst the healthcare workers in the US. And the main drivers are concerns with safety and serious side effects. In order for mRNA to improve its translation, especially beyond COVID, these concerns around them, especially around safety, need to be addressed. And the overall broader context of this is that COVID-19 has shown us the economic impact of diseases, the havoc that they can wreak on entire societies, but it's also shown us the power of vaccines in tampering the spread of viruses and also returning us to a sense of normalcy by saving lives. As a result, it's clear that preventing future pandemics and controlling disease spread will be vital. mRNA vaccines have been a huge success. A little over 40 days to the clinic and under a year to emergency use authorization, they've saved millions of lives, and they continue to save lives every day. But from the slide I just showed you, you can see that there's still challenges with mRNA vaccines. First, let me clarify what exactly an mRNA vaccine is. When you hear it, you're actually thinking of two things. The first is the mRNA, which encodes the antigen, for example, the spike protein for COVID. But for it to actually work, it needs to get into the cells of your body. And this is where lipid nanoparticles come in. And most of these adverse effects and allergic reactions you hear about come from these lipid nanoparticles. And so mRNA vaccines face challenges in their delivery modality, mainly due to LNPs in terms of safety, costs, manufacturing, and more. A common phrase we've heard is that while mRNA vaccines may be tolerable during a pandemic, they may not be tolerable after this one. And so clearly with the power that they have, we need to improve their delivery. And so the first question is, is there another way to deliver mRNA? Electroporation is one such method. The usage of an electric field to produce pores in a cell membrane and allow DNA, RNA, or eventually any biologic to enter. But as you can see from the top right, this has been limited because these devices are expensive, they're bulky, they're difficult to produce, and even difficult to use. But what if we could solve each of these problems? Then let's take this a step further. What if mRNA was a plug-and-play platform? Before you actually get your shot, consider what exactly that mRNA vaccine has gone through, and it's actually a long and arduous process. First, you need to identify the target, optimize your sequence, construct it, and then the process with LNPs begins. You need to design and produce them, screen candidates until you can find one that'll work, formulate the mRNA into the LNP, characterize it, come up with a process to scale, and then finally get it into those vials. But what if we could avoid all this? Imagine no screening, no formulating, and have a one-size-fits-all solution where you could simply insert mRNA into a cartridge and deliver your treatment. Imagine the power and efficacy of LNPs, but improved safety, simplicity, cost, and effectiveness. And that is the solution we're proposing here. Imagine an mRNA free of LNPs. ePatch is our technology. It's an inexpensive, portable, and battery-free electroporation system that can deliver mRNA vaccines without lipid nanoparticles and traditional needles. Initial benchmarking shows that it provides comparable efficacy and improved safety relative to commercial electroporators, and that's expected against lipid nanoparticles. It is minimally invasive, designed to be single-dose units that can be administered by minimally trained personnel in rapid fashion. Another challenge with lipid nanoparticles is their manufacturing. It's one of the main reasons why mRNA is only produced in certain countries and why other countries are slow to adopt it. It's simply difficult, complex, and very technical to produce. In contrast, our devices don't require access to external power, and they're produced using existing infrastructure, and it allows for scalability, not just in the millions, but to the billions of units in a one-size-fits-all manner. To give you a broader context on the overall market, The market for human vaccines is valued at $35 billion. We've subsegmented that into $19 billion focused on transdermal drug delivery. And we believe our near-term market opportunity is $5 billion, focusing on two buckets, which are diseases where there are no approved vaccines, and diseases that cause pandemics, such as influenza and now coronaviruses. But of course, electroporation is not a new technology. It's been around since the early 1980s, and there are many companies that are working on such systems. But only one of them is actually approved for use, and it's in oncology in Europe. But these systems still succumb to the same limitations I mentioned earlier. They're bulky, they're expensive to produce, and they're difficult to scale. And these are the primary issues that we address. And so it's important to consider the competitive landscape of where we fit in. And we believe we address the limitations with these electroporation systems. Taking it a step further, across our over 100 customer discovery interviews we conducted with folks across pharmaceutical companies, we were able to identify a key set of value propositions that ePatch provides relative to lipid nanoparticles and commercial electroporators. Against lipid nanoparticles, the main advantages are improved safety, comparable efficacy, a much cheaper cost to produce, a simpler process to manufacture and scale, safe delivery of large cargoes, and the potential for a one-dose mRNA vaccine instead of 2 shots, improving its accessibility and allowing for patients to not have to return for their second dose. Now considering how do we plan on translating this technology and what do we envision into the future? And we are looking at ePatch in 2 distinct segments. The first is for the United States, Europe, and developed countries across the world, where we plan on partnering with companies that are developing mRNA and DNA vaccines in exchange for royalties and milestones payments for our IPs. The second is to focus on places where mRNA vaccines are not the primary vaccine of choice. Due to logistical issues, cost issues, manufacturing issues, etc., mRNA vaccines aren't as widely accessible as we find here in the United States. And we hope to be able to get mRNA vaccines out to the last mile to every person that needs them. And so we want to work with nonprofits and NGOs in exchange for grants and non-dilutive funding and co-develop mRNA vaccines with them to hopefully vaccinate the billions who don't have access at the moment. From an intellectual property standpoint, we are currently protected by strong IP for nucleic acid therapeutics. And from a regulatory standpoint, we're regulated as a combination drug device system. And we've spoken to regulatory experts who advised us on the process and have also told us that given that there are over 50 clinical trials in electroporation at the moment, it should aid in our overall approval process. Considering the progress that we've made to date, we've completed a DNA vaccine study in small animals, and the results of which will be published in the coming weeks. We have an ongoing study with an mRNA vaccine in small animals, and our technology is currently in an advanced prototype stage, which is suitable for animal research, and we're currently seeking and working with manufacturing partners to scale this to MVPs that can be used in the regulatory approval process, as well as in clinical trials. We've conducted over 100 customer interviews with key decision makers and all the way down to scientists at these organizations. And many of them have expressed interest in potential collaborations, including on mRNA vaccines and some on DNA vaccines. And finally, our core founding team has been established with strong academic collaborators across Georgia Tech and Emory for continued research and expansion. Finally, I'd like to consider what exactly is the future for ePatch and what exactly is possible. In the short term, we plan on focusing on mRNA vaccines, addressing these limitations with lipid nanoparticles to make mRNA vaccines accessible and affordable to the wider population. But in the long term, we hope to broaden ePatch and leverage it as a platform technology into other applications of electroporation, such as monoclonal antibodies, protein therapies, immunotherapies, and localized gene editing. Imagine a new approach where you could screen targets on site, synthesize the mRNA right there, and deliver it into a patient. A whole new world of personalized medicine. Our long-term vision is to enable the next generation of biomedicine and transform the delivery of mRNA therapeutics. And we plan to start this by beginning with mRNA vaccines, making them more affordable, more accessible, and more scalable by addressing the limitations with lipid nanoparticles and addressing the safety concerns in those patient stories, and ultimately impacting and saving lives in the future. We're currently seeking industry collaborators who are working in the mRNA and DNA vaccine space and also nucleic acid therapeutics more broadly. If you'd like to work with us or have any questions or would like more information, please feel free to reach out to me. I've included my email address here, and also can connect with me on LinkedIn. And I'm happy to answer more questions and hope to work together in the near future. Thank you very much for listening to my presentation, and have a wonderful day.